Analysis of restriction enzyme sites. Restriction sites where restriction enzyme cut DNA sequences can be made by cutting DNA with selected restriction enzymes, electrophoresing the DNA on agarose gel, and visualizing the DNA binding pattern with ethylene bromide. Mapping the positions of restriction site generate a linkage map that can have many applications: forensic, systematics, population genetics. DNA is cut with different enzymes, and each DNA enzyme mixture is loaded on a separate lag in the gel. Negatively charged DNA separately by size in the electric field. Fragment pattern produced by the gel stained with ethylene bromide is photographed or imaged and processed by software. Distance each band migrate is calibrated with known size standard of pre-cut DNA, for example ladders. Resulting pattern of different number and size of fragment cut by different enzyme is interpreted to make restriction site map. In these bands, small band migrate faster. Many copies of colon linear DNA fragments. Analyze DNA by restriction enzyme. Several types of restriction enzymes: cleaved and agarose gel electrophoresis. Here is agarose gel electrophoresis. Longer DNAs, migration, short DNAs. The results: interpretation, construct models, conclusion. Application of restriction enzyme site analysis. Analyze gene structure with large and fine scale mapping, limited by number of restriction sites in the target sequence and the variety of enzymes that are restricted for analysis. Restriction fragment length polymorphism can be used for forensic polygenetic analysis. Due to variation in the number and the composition of different restriction sites, less expensive than DNA sequencing, cut DNA can be probed without cloning using the Southern plot, which can be used to determine if the gene share homology with a proof and how many copies of gene might exist. Southern plot, invented by Edward Tabor after restriction enzyme cutting and electrophoresis. Genomic DNA appear as a continuous smear on agarose. Denature DNA in the gel with alkaline solution. Neutralize gel and place on blotting paper. Place hybridization membrane over the gel. Buffer solution is waked up by blotting paper. Fragment on the filter are arranged the same as on the gel. Saturate the membrane with labeled probe. The component from top to bottom. Cellular DNA. Restriction fragment of length determined by location of recognition sequence for restriction enzyme. Gel electrophoresis. Transfer to membrane layer filter by Southern plot technique. DNA fragment transferred exactly as they were arranged in the agarose gel. DNA fragments complementary to the probe are visible after autoradiography or chemiluminescence. Northern plot similar to the Southern plot but used to probe the RNA, not DNA. Can be used to determine the single RNA size, detect the differences in the promoter and terminator sites. Can be used to determine if a particular gene is expressed, and if so, how many, what tissue type, and we name the life cycle.